So there are three types of transport for molecules to go across the cell membrane. The first type is called passive transport, and within passive transport, it's made up of either diffusion, for example, osmosis is a type of diffusion, or facilitated diffusion. The second type of transport is called active transport. An example of that would be the sodium potassium pump. And the third type of transport is called vesicle transport, like endocytosis or exocytosis. Today we're going to talk about passive transport. They do not use any energy. The molecules move randomly, and molecules spread out from high to low. So here are the three different types of passive transport that we see. The first one is just diffusion. So the purple circles will just pass right through the cell membrane, the phospholipid bilayer. An example would this be water. That's a special type of diffusion. It's basically diffusion of just water, and that's called osmosis. There are other types of diffusion where particles can just pass right through the cell membrane as well. Second type of passive transport is called facilitated diffusion. And here there are two examples. The first one with the orange triangles is just a channel protein, and that just allows a selective transport of one type of hydrophilic, that's a water-loving molecule, across the cell membrane. The second type is called a carrier protein, and this is a little bit more complex, in which the molecule will bond to a receptor site, and then it kind of moves through the cellular membrane. So it's kind of like once it bonds, it closes, and then the other side opens up, and it can kind of go through. The one thing to know is that diffusion, just in general, is the net movement of material from an area of high concentration to an area of low concentration. And the difference of concentration between the two areas is termed or known as the concentration gradient. All right, so two types of passive transport. We have diffusion, for example, osmosis, and that's the diffusion of water. And we have facilitated diffusion, which is the diffusion with the help of transport proteins. Like I mentioned before, a transport protein could be a channel protein or a carrier protein. So diffusion is a random movement from high to low until this equilibrium is reached, and equilibrium is the point in which the concentration is the same on both sides of the membrane. Diffusion of water is known as osmosis, and again, that's from that high to low. So if you look at the image right here, you will see this on this first thing here, the left-hand side, that has a high concentration, meaning it has a lot of solute. In this case, that solute is sugar. And on the right side of that, this has a low concentration. Low solute. And this says a high solute. So in this case, because I mentioned before it goes from high to low. We come back to this slide here. It goes from high to low, and you might think, well, it goes from high to low. That doesn't quite make sense with what direction it goes. But if you remember, okay, here it goes from high to low. But if you recall, this case we are talking about, sorry about that, we're talking just about osmosis. So while this has a high solute, it also has a low concentration of water. And then this on the right hand side, this has a high concentration of water. So it goes from high water concentration to low water concentration. And that's where it goes from the high to low. Because it's a movement of water in this case. All right, so here's another facilitated diffusion. This is just, it's a passive transport, like I mentioned, but now we need the help of a protein, which is this purple thing right here in the cell membrane. And remember, this also goes from high to low. So in this case, the concentration of these red molecules, let's say they're, I don't know, a sugar molecule or something, and this is a high concentration, and this has a low concentration of those red circles. And so they want to move from high to low in this direction. So they'll bond to the protein. They carry a protein at the bonding site, and they'll bond there, and then that will open up this protein and allow it to kind of go through.